Today's lesson is regarding proteins and enzymes. And again, it's the SBI4U. We're looking at proteins today because proteins are such an important factor in life. Because if you look at a cell is made up of approximately 50% dry weight of protein. And we use enzymes for just about, or sorry, not enzymes, we use proteins for just about everything. And if you start looking at it, it's structural. There are enzymes, which are catalysts, which we will talk more about. Storage, transportation, or transport of molecules plus the cells. So if you look at cilia and the flagella, those are basically proteinaceous, and there's defense mechanisms, and yeah, that's pretty much what we're looking at. And remember that the goal of life is to sustain those chemical reactions that we have in our body, and so enzymes are key to almost every chemical reaction in our body. We have approximately, well, there's tens of thousands, so I'm just going to say plus 10,000 different proteins in our body. In our bodies. Okay? We're talking humans, we're not talking all the other organisms that also have proteins and enzymes, but we're just looking right now at the human aspect of it. Okay? And if you think about it, a protein, to look at the simple definition of a protein, it is one or more polypeptides sorry about my writing, it's not coming across very well. One or more polypeptides folded and or coiled into a specific 3D or three-dimensional shape. And the vital thing for this aspect right here is each protein has its own shape. Okay, so each protein is unique in its shape, and it's really important to note that that shape and that protein, if they're not the right shape, that protein will not function. And I will say it once, and I'm going to say it many times today, that each protein has its own shape, and shape determines its function. Now we're going to continue and discuss sort of the structure of proteins. And so we can continue on here, and then we're going to go to the next page. Oh my. Oh, stop, pause. 2. And apparently we have to start a new planning. And as we talked about it, remember, a protein is made up of as a polypeptide. So your polymer is your polypeptide. Your monomer, remember we talked about monomers being single units? Well, the polymer is the many units. The monomer is a generic called an amino acid. Okay? And there are 20 different amino acids. Amino acids. And it's those combination of the different amino acids and the polypeptide sequences that actually make up the structure of a protein. Okay? And I'm just going to call it the generic amino acid. Because each amino acid has a general structure to it, and that general structure is a common central carbon. And as we've talked many times before, carbon has 4 bond sites. The amino group, which is the functional group NH2 there, right? So off here, we'll call that the amino end. Okay? And the acid, if you think about an acid in biomolecules, a lot of the acids are the carboxylic acids or the carboxyl group, so that's C, double bond O, OH. So from the central carbon, you build your carboxylic acid off the end, and then every amino acid has a hydrogen, and off from the last one, what makes each amino acid unique, there are different R groups. So for instance, alanine and glycine. Glycine, the R group for glycine is just a hydrogen. For alanine, the R group for alanine is a methyl group. So you would substitute that into that space. And these R groups, so when you look at the R groups, that's what determines the properties of the amino acid and how they behave when they're next to each other, and that's how the shape of proteins exist, is the R groups. Give each amino acid its properties. Now, proteins exist within the polypeptide sequence has a certain order, and that order is what's deemed as the primary structure of a protein. There's 4 levels. There's primary, secondary, tertiary, and quaternary. We'll talk about those in a second, but let's start building the amino acids, okay, or start building the polypeptide sequence. So in order to build a polypeptide sequence, you need 2 amino acids. So I'm going to take glycine, and that is a amino acid. So you've got your NH4, central carbon, hydrogen, hydrogen, because it's glycine, and my carboxylic acid. Then I'm going to mix it with a alanine, which has the methyl group as its R. So again, as you're building it, there's your amino group, there's your central carbon, there's your hydrogen, the R group I'm going to put up here, and then our carboxylic acid group. Okay? And as we talked before, when we add them together, that process we call dehydration, synthesis, also known as condensation reaction. And in dehydration synthesis, we're going to take the water and we're going to remove it. So right here, see there's an HOH. We're going to remove that, and we're going to make some water. And what you're left with at the end of the reaction is water because you made it, plus, now if we sketch this out, remember there's a central carbon with a double bonded O, right? That's the oxygen. And then we have the carbon directly bonding with the nitrogen. And we get CH3 with the hydrogen, C double bond O, OH. And so now you have a dipeptide formed, and the bond between the carbon and the nitrogen is called a peptide bond.